So our next talk is going to be by Dr. Nathan Price. Nathan is an associate professor at the Institute for System Biology and affiliate associate professor at the University of Washington. He did his bachelor's in chemical engineering at Brigham Young and his PhD at the University of California, San Diego. And I'm really excited to hear Nathan talk today because I think this morning's session has really highlighted the value of omic technologies in producing these molecular signatures and taking a chunk out of this chemical information content in the cell. The real challenge, though, I think that we face going forward is how do we integrate this information and somehow reduce the volumes of this chemical information to the bare essence of the specific signaling networks or pathways that make sense. And this is an incredibly complex informatic challenge. So with that, I'm going to hand it over to Nathan and let him tell us, hopefully, how we might do this, or at least take a stab at it.
Well, thank you, Mike, for that kind introduction. I'd like to thank the organizers for inviting me. Have quite a number of relationships here at Ohio State, primarily with Clay Marsh as part of a P4 Medicine Institute that he and I are both on the board for. And so has been really great to be able to come out here and interact with a number of people from time to time. So I'm going to talk a little bit about systems approaches to molecular diagnostics. So I'm going to start, although I don't really need to go through this, it's already been shown, and actually with some slides I actually like a little better than this one. But basically we have, as we know, two exponentially rising curves that we're all sort of sitting on top. We have Moore's Law, which is the slow one for computing, and we have the increase in genomics, which is going, of course, massively faster. So this leads naturally to lots of optimism in terms of the potential of the future, and I share very much in that optimism. As was already highlighted this morning, we also have not seen an exponential rise yet in terms of outputs. This was brought forth very nicely in our first talk this morning of the sort of flatlining of therapeutics. Here I'm showing blood diagnostics, which is the part that's shown here. And you can see that over the past couple of decades, we get between 1, a high of 4, but essentially, again, fairly flat. So this is a big issue for the omics community, which is how do we take all of the amazing advances, which are accumulating, which are going to have tremendous impact, but how do we get there? How do we start translating all of this great information that we're generating into tests that can be effective in the clinic? This was an issue that was studied by the Institute of Medicine recently, and I was actually a member of that committee. But we basically spent the last 18 months evaluating the state of omics research and this issue of why there are so many papers in omics and so few tests so far that have made it throughout. And I'm going to talk about some of the issues associated with that today. For those of you who are in this field, I definitely recommend picking up the report. I think there's some interesting things to go through. Should mention also that this has to do a lot with issues also relating to data reproducibility. I got an email from yesterday inviting me to a workshop at NCI that Harold Varmus is putting together on data reproducibility, which is in response to some issues that a number of people here are probably aware of, where pharmaceutical companies claim that they have taken many omics studies from, I think they took 40 or 50 from high throughput papers and weren't able to get what they call data reproducibility. So one of the issues that's really before us as a community now is, well, what does it mean to be reproducible in high throughput omics? That's actually not a obvious thing, because for statistical considerations, when you're in the small sample size regime, you do not expect one study to reproduce exactly another. And so there's a whole host of issues that come into play as we think about how do we take all this tremendously exciting new technologies and start translating that to something that will have an effect. Okay, so I'm going to start with a really simple story. Just because I think it will lay out some of the fundamentals of what I want to talk about as we go forward. And it's going to introduce also the notion of a relative expression reversal. which is something that I'm going to use quite a bit as a basic component of some of the methods I'm going to talk about today. Okay, so this was a study that I did several years ago in collaboration with Wei Zhang, who is a professor at MD Anderson Cancer Center. And what we set out to do here was to come up with a diagnostic signature for a very specific problem. And in this case, it was to differentiate two cancers that had, until fairly recently at that point, been thought to be the same cancer, gastrointestinal stromal tumors and leiomyosarcomas. We now understand those to be separate diseases. And they're very separate diseases in an important way because they require different treatments. Gastrointestinal stromal tumor is quite responsive to Gleevec, but with about 50 to 80% of patients responding very favorably to Gleevec treatment. But it's not very, it's very recalcitrant against cytotoxic chemotherapy. Whereas LMS, you can treat quite well with cytotoxic chemotherapy, but it is non-responsive to Gleevec. So what you have are two cancers that morphologically look very, very similar, but require very different treatments. So the question was, could we have a simple omics test that would tell us which is which? And so when I set about doing this, I did this in a very simple sort of way, I would say. And this was based on an approach that was developed by by a good friend and collaborator, Don Gieman, at Johns Hopkins. And basically, we're going to introduce here the notion of a relative expression reversal. And this is a simple concept. Basically, it means we're going to try to find a classification rule of the following type. If gene A is more highly expressed than gene B, that's going to be a vote for class 1, GIST. Otherwise, class 2, LMS. Okay? So that's what I mean by a relative expression reversal. Just two genes that are where the one gene is more highly expressed in one phenotype and it reverses in the second phenotype. Okay, and then you can combine these in lots of interesting ways and do voting and such. So why would we do this? So it actually has a number of different advantages. For one, it does not vary with data normalization techniques that do not shuffle the rank ordering of genes. So for example, normalizing to total RNA doesn't matter because it already has its own internal standard within every sample of just comparing these two genes. So that makes it quite simple. The nice thing is when you look at markers in pairs like this, since you sort of have your own internal standard, you don't have to learn a population-wide cutoff like you would, say, with a single marker. Its reported accuracies in the literature are actually comparable to support vector machines or pattern analysis of microarrays or any other state-of-the-art classification methods. And it's designed this simply because it's a means to avoid overfitting. Okay, so in the case that I'll show you here, in essence, you're looking at how many pairs can you evaluate. The model is already fixed. You're only going to look at greater than or less than. And so as long as n choose 2, where n is the number of genes, is much less than 2 to the m, where m is the number of samples, right? So the total number of permutations that you're searching. If you find something with this sort of simple approach that works, it's likely not to be due to overfitting. Okay, so we applied this, and here is the pair that came out. So this was to separate GIST and LMS. Again, this was done in collaboration with MD Anderson Cancer Center and Wei Zhang. So here we have, so every X here corresponds to someone who had a GIST tumor. The red circles for LMS. And then the line here is not a fit to the data. It just has a slope of 1, intercept 0. Just represents greater than or less than. And it turns out that if you compare these two genes, one of which was an uncharacterized open reading frame, chromosome 9 ORF65, which has since been named PRUNE2. And the other was aptly named obscurin. Not a lot known about it either at the time. But in essence, if obscurin is more highly expressed than C9 ORF65, you essentially always have a GIST. And if it's the opposite, you essentially always have an LMS. Now, of course, you can't trust data that you see that just comes out of this. Now, as I mentioned in the previous slide, or showed on the previous slide, we evaluate about 1 billion different possible pairs to come up with this. To get a perfect separation, there are 10 to the 20th different possibilities. So you're not likely to see this by chance. If we do this in cross-validation, we get an estimated accuracy of 98%. Now, cross-validation has a number of issues, and I'm going to talk about a lot of those issues in the next segment. But one of the issues that it has is it has high variance, which means that in any particular study, your estimate of how good your classifier is can vary significantly. So what you really want to do is to go out and get an independent test set. So that's what we did. We got all the available new samples that were collected at MD Anderson, which wasn't a huge number. We were able to get 19 additional samples, and we also took 20 samples from the microarray study, put those together on a new platform using RT-PCR, and the test achieved 100% accuracy in the new set. And so this was a blinded test. And so basically, we've got good reproducibility here. It was sort of a simple study, but at least this recapitulated for us very well the diagnosis that was done at MD Anderson. And so, in principle, can replace a battery of tests that are used to do the diagnosis today. And this can be done also from very small amounts of tissue. Okay. So just a few general lessons. So I introduced this notion of the relative expression reversals. Okay, and I'm going to come back to that, which are just these simple pair reversals where you convert from an analog signal into a digital, because now we just have a 1 or a 0 output in terms of two markers. These are very easy to implement clinically because you can just do RT-PCR on a few genes, or if you're doing proteomics, you can just use an ELISA or something simple like this. And so this is an approach that we're quite interested in. Okay, so in my lab, we've been developing a wide variety of computational tools that we can apply to analysis of high-throughput data. And I'll just mention a few of them here, mostly just by way of making you aware of them. So this top-scoring pairs approach, which we can also expand into top-scoring triples, we now have implemented for GPUs, which are essentially what's in your Xbox graphic processing units. We can make this algorithm 1,000 times faster, which means that we can do it at human scale very nicely now. We have an approach that generalizes this then to not just pairs or triples, but sort of any number of markers that you want. I'm going to talk about two in detail, so I won't belabor them here. I'm going to talk to you about Isaac and Dirac in the next two sections. We have a software package that will be coming out that will enable these to happen. And then in work that I actually was not able to fit in today, but I want to just mention it in case it's useful, we published in PNAS in 2010 a new method for integrating metabolic and regulatory network models, which is a way to bridge statistical regulatory network models with biochemical models that are detailed in terms of metabolism. So we're very interested in applying this going forward to having metabolism-based signatures that integrate across transcriptome, proteome, and metabolome. And then we also have a method which we published in PNAS in 2011, the next year, on something called Asterix, which is a gene which identifies gene regulatory network influences, which we applied to getting the first transcriptional regulatory network influence model for a brain, which we did for the honeybee. Okay, so anyway, just to give you a sense, all those are available if you're interested in our download page. Okay, so that was a simple study meant to, by way of introduction. So now what I want to do is to come back to some of the big issues that I alluded to before in terms of reproducibility and in terms of thinking more broadly about signatures and why we run into problems. Okay, so one of the things that we're really interested in at ISB, and that's been advocated and pioneered by Lee Hood for many years, is an envisioned future for the use of blood diagnostics, okay, schematically shown here. And basically, this is a major effort at the Institute for Systems Biology. We have a major partnership with the government of Luxembourg, which has given funding to ISB of $100 million over 5 years to essentially half of which is to implement blood diagnostics, half of which to do personalized genomics. But basically, so what we're trying to do here is to come up with organ-specific signatures where you have a set of proteins, for example, in the blood, that are specific to the brain, that are only or largely made in the brain or in the lung or in the liver, and so forth, and get all that information out of the blood, and then come up with ways to integrate or pull out signal associated with all of these signals that are being put together in the blood from all of these different tissues. Now, if you start thinking about this as a computational problem, which I do a lot, you can imagine that this is extremely daunting because you have tumors when they begin are very small, right? So you have small amounts of protein that are coming into the blood, and you want to try to be able to deconvolute these signals. The other issue that's a major issue is that if you're thinking about doing blood diagnostics like this that are going to be predictive, right, that you're going to have tests, you're essentially going to monitor blood every 6 months in patients looking for early warning signs of disease, that's very, very different than the situation I just told you about in the last one. So the last one worked quite well, but that was very much downstream. We have GIST or LMS, it's one of those two. We want to do a final separation, okay, very specific on the tissue. Here, the issues become much, much more complicated because it's not one disease versus one control. It could be any disease or any environmental perturbation, and you're trying to differentiate what is normal from what is not normal, okay? This is clearly not a solvable problem today broadly, right? I think it's extremely exciting. There are tons and tons of potential here, but this is really, really challenging, okay? So this, I'm mentioning this just to sort of orient you to the goal of where we're trying to get to. So the envisioned way that these get put together then is to think about wellness, right? Separate, do you have a disease or do you not, right? That's the first question. Are you healthy? What are metrics of wellness? Organ-specific parameters for each of the different organs. And then once you determine from the organ-specific signatures that you have a disease in a particular organ, right? You see maybe the brain-specific transcripts are, the brain-specific proteins are changing a lot. Then you get into all of the issues to separate out the different diseases. Are you early stage or late stage? What therapy should you have, et cetera? Okay, so very, very challenging. And so this is just some of the things that we have to think about as a community. How are we going to deal with predictive medicine in this complex kind of environment? Okay, so I'm going to talk to you today about something that's a little bit simpler, but that we took on to start thinking about some of the issues associated with that vision that I just talked to you about. So here we're going to do this from transcriptomics because it's really the only data source which is abundant enough right now. But we're going to look broadly across all of the different major diseases of an organ system. Okay. So why might we do this? So one, I mentioned the issue, certainly in the blood, of organ specificity being very important. Right? We want to be able to narrow down, where is the most likely source of a particular perturbation. I talked about this hierarchy of classification. You start with context-independent classifiers. So a context-independent classifier is like the case of the blood, meaning we're going to measure your blood every 6 months. We want to say something, in terms of your health state. That means that you don't know a lot coming in, right? You need to be able to differentiate many, many different phenotypes at the same time. And then there's a winnowing down to what I'll call context-dependent classifiers. That is the example I gave for GIST and LMS, right? You already know a lot. You've been to the doctor. You've cut out your tumor. You're just trying to figure out which treatment do you need, right? So there, a lot of context has already been generated. So you're really doing a course-define type of classification problem. Okay, the other issues that we're thinking about here is one of the big problems that arises in signatures is that when most people set out to do studies early stage, they're not thinking very much about the end stage. Okay, and this was brought up beautifully in Dr. Austin's talk at the beginning of the day. So in essence, when an investigator sets out to do a study, typically, you are hoping that it has that relevance downstream, but the other more proximal issue is to get something that's publishable, right? And so what we often do, and this is a perfectly reasonable thing to do, I do this, but it's the perfectly reasonable thing to do, but what it leads to is that typically you'll have a case and a control, and you try to get everything standardized as well as possible so that we can get a signal out about what is different, right? So if we can get a good signal from a case and control, that's great. But the problem is that when you go out into the real world, there's all these other sources of variance that can then change the parameters that we're using to separate that nice, carefully controlled case and control that we have. So when we do that, this is one of the reasons why a lot of these signatures will then fail as they go forward. So one of the things that, that I'm a big proponent of is that if we're really thinking about the end game in terms of developing diagnostics and therapies, we want to make the problem as hard as we can up front so that that death valley, right, that Dr. Austin talked about is not quite so steep. So that's one of the issues that we're trying to get at here, which is rather than trying to come up with a signature that separates, say, glioblastoma from normal, we're going to set up a hierarchy or a structured signature that has to tell the difference between many diseases of the brain simultaneously. Okay, so we're making the background much more complex, hoping to find signatures that are much more specific to our particular context. So this was just, again, as I mentioned, transcriptomic data, which we're using sort of as a proxy. We'd rather have blood data here, but you know, this is what is really available a lot now. So this is just a multidimensional scaling plot to show the variances. There are about 1,000 samples in here. And what you'll notice, and I'm going to come back to this a lot, so this that's labeled here A1, which is a grade 1 astrocytoma, really a pilocytic astrocytoma is what it really is. But basically we have a pilocytic astrocytoma here, and you can see a set of red balls here, and you can see another set of red balls up here. Say, well, what's the difference between those? They were measured by different labs. Okay, so this is another really big confounding factor, is that when we have a signature in the literature that says this is a separation from pilocytic astrocytoma in normal, that does not hold up generally very well as you move from one lab to another. I'm going to talk a lot about that here just in a minute. Okay, just by quick way of introduction, I don't want to go through this in any great detail, but this is a new method that we've developed, which we call ISAC, which is the identification of structured signatures and classifiers. This is a way that we use to do the organ-specific structured signatures that I'll be talking about here. And it's basically an approach that builds on that relative expression reversal type of idea. At each one of the nodes, we come up with a set of relative expression reversals, in this case for genes. You get a 1 if this relationship's true, a 0 if it's false, and then you do a vote amongst them to decide whether or not you likely have one of these, if we're looking at a transcriptome that comes from one of these diseases or not. And then we do that at every node. So this is not a decision tree in the sense that when you come down, you can get a yes vote on both of these, you can get a no vote on both of these, and so the specificity comes from the fact you have to get a yes vote all the way down to a leaf in order to make the diagnosis. And then we have tie-breaking procedures if it goes down multiple at the same time. Okay, and we do that also because in the real clinical setting, it's possible you have both, right? And so, so there are ways to think about that. Okay, so I don't want to belabor the method too much. But so we applied that to the transcriptomics data, okay? So here we have a signature. It has to, for every sample that comes in, decide whether or not it should be in the tree at all or not, and then make the diagnosis amongst several different possible diseases. Pilocytic astrocytoma, Alzheimer's, glioblastoma, medulloblastoma, meningioma, neuroblastoma, oligodendroglioma, Parkinson's, or normal control. Okay. And so when we do this, we get quite good accuracy. We get 94% accuracy. This is done in 10-fold cross-validation. Okay, so you learn the entirety of the structure. This takes quite a lot of computational time, so we learn the entirety of the structure, and then we leave out 10%, right? Then we get the accuracy on the 10%, do that 10 times, right? Standard 10-fold cross-validation. If we do it just on the brain cancers, right, which gives us a more homogeneous set of sample types, which is important, this accuracy is 90%. So this is just in terms of taking a tumor out of the brain, run it through this classifier. It's based on 50 genes. Those 50 genes can, with 90% accuracy, tell you which of the major brain cancers you have simultaneously. And remember here, right, when you're doing binary comparisons, 50% is random guess. Right, as you add up, that of course falls. Here 15% or so is random guess. Okay. If we restrict ourselves then to genes that are only secreted into the blood, okay, so now we're looking at proxies for something that we hope will be able to translate into a signal. So this is for genes that encode for proteins that have been identified in the blood. There are about 3,000 that we looked at here. This drops a little bit down to 87%. Okay, so that's just 3 different ways to sort of slice this data to think about. Okay, so pretty good. So I'd say that's pretty positive. So we were quite happy with the outcome where we could get around 90% accuracy from such a diverse set of transcriptomes. And again, of course, in a medical setting, you don't have to limit yourself only to transcriptome. You also have symptoms. You also have morphology. There's all kinds of other things you can bring to bear. Okay, so that's the good news. So now let's talk a little bit about the bad news, the challenges, and why some of these things in the community really haven't translates. So I'm going to introduce this notion of lab effects. So let's come back. We're going to use this Isaac structured signature again. We're going to relearn it, but this time we're going to do the error estimation in a different way. So before I left out 10% of the samples and did standard cross-validation as is typically done. Here, I'm going to do something very different. I'm going to leave out one study at a time. So I'll take, one study, will be in the training set, say from UCLA, and then we'll have a test set, say done from here, and so forth. So we're going to leave out one study at a time. Now, when you leave out one study, there's a lot of variables that aren't being controlled. So in some ways, it's not really a fair comparison of, say, the classification method. It's really not. The tenfold cross-validation is. But this is a fair measure of the heterogeneity that's really out there when you move from one site to another. Okay? Here's what happens. Okay, so this takes a lot of computational time because we have to relearn from scratch every single step, and I'm really a stickler on that in my group. So every particular step has to be redone from scratch. When you leave the one study out, you relearn the entirety of the whole brain-specific classifier, and then you test it on the one left out. Then you leave out another study, you do the whole thing again. This takes about on the order of a month on our 100-node cluster to run through all this. So it's a lot of computation that goes behind this. Okay, so here, so what this means, so every bar you see here, this means that this is the study, this epengioma study was left out. This is the accuracy. Now before, we got near 100% accuracy on this. So if we learn across the two and we leave out a random sample, we do very well. If we learn on one and test on the other, we get essentially random guess. Okay, and I'm going to come back to this. The key point here is that the differences between what are measured in labs are big, and there's all kinds of reasons for this. You can see this. Look at this one. This is terrible. So here we have oligodendroglioma. So we have an oligodendroglioma. We get 0%. 0%. That's bad, right? That's worse than random guess. Why do we get 0%? Because the same lab measured one of the glioblastomas. And so we get very high accuracy in picking out the lab that made the measurement, more so than we get the phenotype that was measured. Okay, and that's not just us. This is other people have seen this as well. There is a big silver lining here, though, that I'm really encouraged about. So if you look at glioblastoma, so glioblastoma overall is one of the ones that we did sort of the poorest on. In the first case that I went through, it was about 84% accurate in that range. If you look here, but here we have 5 different labs have measured glioblastoma transcriptomes. And if we take the average of how we do when we leave each of the labs out, turns out to be almost exactly the same as if we do cross-validation. So the nice aspect here is as you start to aggregate across multiple labs, the signal from the phenotype gets amplified. The signal from the lab effects, which can be both technical or biological, starts to be damped. Yeah, question?
When you leave a lab out, do you leave all the studies for that lab or just that lab and for one phenotype?
We've done it both ways, I should say. So in this particular graph, we just left out the one phenotype from the lab. In other studies, because of this kind of confounding effect, I actually had my student go back and say, well, let's just leave out the whole study, both sides, which works a little bit better. But I still talk about this one just because I think it's interesting how well it picks up the lab. But I'll talk about it. Yeah, was there another question?
Well, when you mean 18 GSA data sets, how many patients do you have actually? How many microarrays?
This in total is just under 1,000. Yeah, just under 1,000.
Sorry.
Yes.
I know, for example, this is the case in cancers or in other diseases. What do you consider normal? Because it's hard to get normal patients' tissue as
Normal is really hard. In fact, in about 3 slides from now, I'm going to show normal. Normal is by far our most variant phenotype. In other words, the normals, it's really hard to establish normal. So we do sudden death autopsy like most people do. We take that. We've actually used a variety of different sources for normal. So we've been, as we've chosen to do this, we've wanted to make it as complex a problem as possible. So we take lots of different normals to be in the background, and so we should be able to tell the difference between them, even if they're sudden death, autopsy, or if they're a margin or something like this. We're a little careful with margins because then they really start looking like the phenotype that they are matched with. So it's an excellent point. Normal is the trickiest of all the phenotypes. Yeah, for sure. Yeah, good question. Okay, so we wanted to look at this in a little bit more depth, but a little bit more narrowly. So we went through the cancer literature and we tried to find 2 data sets that were as close to the same as possible. In other words, we just tried to find 2 places that had published that were just really, really similar. Okay, and this is the one that we found. So we took this from PLOS One. Here, this is a data set deposited in Geo. It's 25 healthy tissues, 35 carcinomas. These are colon carcinomas. They did as their initial pre-processing step something called GCRMA. Okay, so this is, for people not in the field, a very technical detail, but it's going to be really relevant here, which is that when you have a microarray, you have an image, and then you have to apply some sort of algorithm to turn that image into a number, right, and do a normalization. Okay. This is actually not reported in lots of papers. They don't even report this step a lot of times, what they did, because it's often considered a minor point. But this used something called GCRMA. So we got this other set, colon carcinoma, 24 healthy tissues, 35 carcinomas. We looked at the, and these are not adenocarcinoma or adenoma. Same thing here, not adenocarcinoma or adenoma. So as close as we could, figure out from going through the papers and going through the data, these were really similar. So if we do just simple principal component analysis, we get what we hope, right? We get two clusters, cancer and normal, only we don't. This is lab 1 and lab 2. Cancer and normal is buried in here, and cancer and normal is buried somewhere in here. Okay, so lab 1 versus lab 2. Lab effects bigger than phenotype effects. That's almost always true. I gave a talk on this at a company last year, SomaLogic, run by Larry Gold. Was showing them all this, and they were extremely positive towards it because they said this is exactly what they see when they're going through and doing their markers and their blood diagnosis. Same thing. Lab effect is bigger than phenotype effect very, very often. This is a big challenge. So we took this same data, but now we take all the raw data, all the raw cell files, and we reprocess them all the same way. Okay? And when we reprocess them all the same way, and I apologize, this color doesn't come out very well. That's green. We process them all the same way. We still get lab 1 and lab 2. That's still the bigger effect. But by doing the processing, this first principal component, which I forgot to mention to you, in the initial case, lab effect was 86% of the variance. Right? We only got, well, it was 86% of the variance. Here, we now get the lab effect is still the biggest, 32% of the variance. But beautifully, if you draw a line almost straight across here, this is cancer and normal. So the signal is there. It's just buried underneath. So if you, and this, in fact, as we've talked to various people, this is actually one of the things you can do is you can look at these sort of modes. You just assume the first one is sort of this lab effect. And if you cut here, you get a very nice diagnostic marker for cancer versus normal. Yeah?
One used GCRMA. What did lab 2 use?
They used Pleon.
So in terms of methodology, was that the sole difference in their methodology?
As far as we could tell. So there's always some differences in these studies because, there's always, even if it's reported in a paper that things are done in the same way, right? Technicians do things a little bit differently, right? There's always technical variance. And there's also biological variance. The population, say, that comes to Ohio State Medical Center, is different than the population that, say, goes to MD Anderson. Or, there's subtle differences just in terms of.
I just mean in terms of like sample preparation prior to the array.
Yeah, as far as we could tell. I mean, they were reported to be basically the same way. So you can do this, of course, over and over again. You're bringing up really important points. If these samples are prepared in different ways, wow, they're hugely different. And so you can start to see the scale of the challenge that we're dealing with. Because when you're measuring an array, it's responsive to all kinds of different things, not just the disease perturbation. It's not just cancer and normal. It's also affected by the biology of the people that are there. Their typical genomic backgrounds, right, which we know are somewhat regional dependent on average. The microbiome, right, which is probably even more strongly regionally dependent. And on and on, the exact methods that are used. I should mention in the study I mentioned for the brain diseases, we do go back to the cell files. We reprocess everything from scratch exactly the same way to try to deal with at least that aspect of variance. That's an aspect of variance we can deal with easily. There are many, many more that we cannot deal with. And this is what's really bogging down the community a lot. Yeah, question?
The separation between different or not different samples, what do you take as p-value or threshold, like two-fold different 1.5?
When we determine, I'm sorry, when we determine what?
So when you consider representative group of genes, the gene signature, what would be your minimum threshold for selecting that gene different from the controls? Is it like, let's say, 1.5, threefold?
Oh, you mean in the gene pairs?
Right, no. So basically you have a set of microarrays,
Yes.
and you have a set of genes that would be common or different, right, between controls. What is the threshold that you set for that gene signature in terms of p-value?
Wait, I thought this is an unsupervised PCA is what's being done?
What I'm showing here is just an unsupervised PCA, yeah. Is that the question? Right, this is unsupervised PCA. There's nothing, the only thing that's happening here is just the variance. We just plot.
How do you select the genes? Like, you select the genes, right?
This uses all the genes, this one. So we don't do any filtering of genes here. This was just PCA on the transcriptomes for this particular slide. Is that the question?
What I'm about is asking from my standpoint, how can I use the information? That's why, because with Dr. Kwang, we work on something similar at the end result, like yours. So I just wanted to know how to separate the controls versus disease, and that was my question basically. How to do that.
You mean the classification method?
Dr. Kwang to answer.
Yeah, I think she was talking about basically when you do comparative analysis.
Well, we
in
do
this case,
compare.
it's more like just a global overview and then there's,
Right, so when we do the comparative analysis, say, like Isaac, I guess is what you're asking. So you feed in a sample and then it tells you, an output of what is the likely diagnosis. So we have an accuracy and a false discovery rate associated with each of those outputs. So it would tell you back, we think that this is, the diagnosis, we think there's a 5% chance that it's wrong, right, the false discovery rate. That's essentially what you would get back, right? And the false discovery rate is, of course, developed based on the p-values. Okay. All right, so just to show this a little bit different way, and I won't go into all the details here. This was done for lung diseases, but this, I think, is a really nice way to visualize the data. So here, we did exactly the same thing that we did in the brain study. This is, again, leaving one lab out at a time. And in this particular case, we did leave out the whole lab, both phenotypes, and then put them back in. So here, what we do. So here, the solid line tells you the accuracy in tenfold cross-validation, and the dashed line tells you what the accuracy is if you leave one lab out at a time. Okay, so what you see here, so for adenocarcinoma, for which there are like 14 different studies, it's essentially the same. So in this sense, by having all these transcriptomes across various different sources, we now see no more difference between whether or not we leave out a particular lab at a time or if we do tenfold cross-validation. This means that the signal has been amplified such that the lab effects really aren't degrading accuracy anymore. Okay? And we've done, I don't have the slides here, we've done this though where we've started with just 2 or 3 or 4, and you can just see the gap go away. Right? It starts, the lab effects are a big issue, they go away, and then when you sort of hit that, it means that you now have something that's much more stable. If you look at asthma, this is where also sample prep kind of techniques come into a play as well. So you see something like asthma, you have this huge gap between cross-validation. Yeah, you can always find it very well. If you don't do that, if you do lab effects, there's this big gap. And the interesting thing we wanted to show here was just that if we use our method, Isaac, or if we use a support vector machine, a multi-class support vector machine approach, qualitatively you get essentially the same answer in terms of where you're at. And normal, which was brought up before, look how that's how we get a huge amount of variance here in terms of normal. And this comes from the fact that normal samples are just come from so many different locations that are very imperfect. Yes?
Time, but the variance depend on regional differences. So do you take into account where the lab is at? Because I mean,
we don't yet.
Environmental and regional would matter a lot.
It does matter a lot. We have not looked at that. So there's a huge amount of work that's gone into this approach, but there's a huge amount more that could be done. There's no doubt about it. We have not looked at regional effects. Would be a great thing to look at, as long as it was statistically well enough powered, which would be my one worry. Yeah, question?
I mean, idea. Maybe what you see is really genetically or epigenetically heterogeneity in the disease. Because like you said,
absolutely, yeah.
Everything coming from the area, so basically, if we speaking about the techniques, why for one cancer it's basically very similar, you couldn't say that all people in the United States studying this type of cancer are accurate and the other are very different in their techniques. Maybe what we see it's really weird.
Yeah, so I hope I've been clear on that point. I am
More variation because
Sure.
type 2 diabetes and obesity would be so much dependent on pollution, genetic factors.
Yeah. So I hope I've been, I guess, not clear enough on this point. I am 100% absolutely not saying that some people do these measurements wrong and some people do them right and that's the reason. Not at all, not by a million miles. What I am saying is that a lot of this is absolutely real. As I've said, it's both technical variance and biological variance, heterogeneity of disease. In fact, so I was invited to give a talk at the NIH to a group in lung genomics. And it was interesting because we had just done this work. And my conclusion out of this, when we saw that these things get saturated, but not at the level we'd really like, right? This is only 65% accuracy. And so, our conclusion was, man, this just looks like, really heterogeneous, or that we've got to think about better ways to define this because we're hit this ceiling and it never really goes above that. And I go to this meeting and like the first sentence, which was with a group of lung experts, like the first sentence out was, lung diseases just really aren't very well molecularly defined. We really need to deal with that. And I thought, oh, great, they're going to like my talk. So, but I think that is an issue. So it's not just technical problems. Technical problems are things that we can deal with and get around, but a lot of it is biological heterogeneity, right? And that's why I was mentioning also the, that there are differences in, microbiome, genome, sequencing, et cetera, that, that can be regional or that are, specific to, family background or whatever, right? We know that there's all these things. That's the point that I think we have to deal with so much in, really getting omics to translate to the real world, which is that there are tons of real biological differences. And when you're doing omics measurements, right, the response of the system doesn't know that it's supposed to be, that it's being tested for phenotype A versus phenotype B. It's going to be responsive to everything that happens to it. And so there's all kinds of reasons that these things change, real biological, technical, and so forth. And so getting it to be reproducible, I think, is the big issue. The other big point that I really want to make here, though, is what I was talking about with, as we start seeing more and more labs accumulate, even if it's heterogeneous at the beginning and we get this big gap between the cross-validation and the individual, when we leave out the individual labs, these things start to come together as you accumulate. So one of the things I think is really powerful is that, as we start having studies that come across, from different locations that are, that have that representative heterogeneity, we start to overcome these effects because, the one that keeps consistently getting amplified is that phenotype that's different. As long as that's really the commonality, that increases it a lot. The reason I think that is really important is that it has strategically a big impact on how we should do these studies going forward, which is that consortiums that are able to leverage data from many different sites are likely to do much better than those that do everything in-house. The reason I think that's important is that for obvious and challenging financial reasons, everybody tends to do these things in-house. So I think from a scientific perspective, at least in terms of amplifying signals, we do this the wrong way. And I made this case to to an executive that Lee Hood and I were meeting with recently at one of the big pharmaceutical companies. And he kind of stopped. He's like, "Wait, are you saying we're doing everything we're doing in biomarkers, and companion biomarkers is wrong?" And I'm like, "Yes." So that's taking it too far, of course, but. But it is an issue. There's things that we do need to think about because it's very easy to overfit. So if we take that, the brain disease data, for example, if we redo that study and we take 50 samples, we base everything on 50 samples from glioblastoma. We do much better if we take the 50 randomly from 4 data sets, and then of course we always test on a 5th data set that's not included at all, compared to taking them from any one of the data sets. You do better in terms of working in a new context if there's some diversity in the sample sets that you have looked at. So I guess that's what I'm saying. Okay, I feel like I just summed up that stuff. Okay. All right, so that's sort of the global view that we've taken of transcriptomics data and looking at things like lab effects. Another issue that we are thinking about is identification of perturbed networks, right? So it's not, I know this is something that a lot of people in the field think about, right? We don't want signatures that are just of any genes, but we'd like them to be related, and we'd like to try to uncover, to the extent we can, different mechanisms that are at play. So we've developed a new approach. This is again based on this concept of relative expression reversals, which we call differential rank conservation, or DRAC. Okay. So essentially what's done here, this is a way of using relative expression values of participating genes as a proxy for the state of a network. Okay. So here we're looking at relative expression reversals. So if this were a particular pathway, for example, all right, and you can have any of these a priori defined pathways that you'd like. You can rank order these from highest expression to lowest expression, and then there's a set of comparisons that can be made, right? Is gene 3 higher than gene 2? Is gene 2 higher than gene 1, et cetera? And you get a binary readout, 1s and 0s, and that represents the relative state of a pathway, okay? So key features of this approach. One, it's again independent of data normalization that doesn't shuffle the ranks of genes. So again, total RNA, don't need to worry about. It's not, doesn't have an effect here. Independent of genes and proteins outside the network. This is, I think, a really nice advantage of this type of approach is that when you ask, is this particular pathway perturbed, you only have to measure the genes or the proteins that are involved in that particular pathway. If you run, for example, gene set enrichment analysis or GO enrichment or anything like that, those are all defined in the context of the other genes, right? In gene set enrichment analysis, you're looking to see if a pack of genes sort of moves up as a group or down in a group in the list, right? And if you do GO enrichment, you're doing Fisher's exact test. So, for example, if you do just the thought experiment, if you had 2 phenotypes in which every gene was differentially expressed, if you run GO enrichment, it tells you that there's no enrichment, right? It would return 0, right? Because nothing is more likely to be enriched than other, which is perfectly valid. Enrichment is different than perturbed, right? So they're distinct concepts. Whereas here, it would return back to you that everything had changed, for example. Okay, and so it's based on sort of a shuffling of the network. Another nice aspect of DRAC is that it provides a distinct mathematical classifier So you get an accuracy back. So you don't just get a p-value, right? A p-value tells you how likely something is to be non-random. If you have lots of samples, you can get a very nice p-value that tells you with absolute certainty that the effect you're looking at is very small, right? And so this gives back an accuracy, which I think is also important. Okay, so there's two different ways that we look at this. So one is you can have pathways or networks that are highly conserved or tightly regulated in a phenotype. That means that this particular network, the rank ordering of its components doesn't change very much. Okay, it's very solid. And you can have some that's loose, loosely regulated or highly variable, and it may change a lot. And then the other thing that we do here is we look at this characteristic ranking of a particular pathway or network as being characteristic of one particular phenotype or another. Okay. Okay, so this we applied then to prostate cancer. And here we were looking at the comparison between normal prostate and metastatic prostate cancer. And so what you get is you have a template then. So for the MAPK network, which we involved about between 15 and 20 genes, and we take this network and we just ask, if all I knew from this sample was the expression of the genes in this network, can I tell the difference between whether or not this is metastatic prostate cancer or not? Okay? So sort of a simple thing. So when we do that, essentially how this works is you get a score that says how similar is the ranking from this particular person's tumor to the characteristic one for metastatic prostate cancer. And then how similar does it look to normal prostate? And you get a score, and then all we do is a comparison. We just say which one's higher. Okay? And so what you see is that because of this comparison, a few things here, like this one here would be an outlier. Right? This is if we just set a threshold. That's what this blue and yellow mean. This is the average or the threshold in the two cases. You can see that there's a nice switch. But when you have metastasis, you get these. This one is picked up because of the comparison, and then the same thing on the other side. You get this one, which we would have missed, but again, it comes out right on the comparison. So if we do the differential, we get this nice signature here for MAP kinase that separates beautifully the metastatic from predicted as normal. And so if we put that in cross-validation, again, to avoid overfitting, we can then take this MAP kinase network and we get 92% accuracy. If we take prostate versus normal, we get 96 for metastatic prostate versus normal, and we can get 96% also for metastatic cancer versus prostate cancer. And our main point with doing DIREC is actually not to have a classification algorithm. It actually works quite well as a classification algorithm, but that wasn't really the point. The point is that we take the classification accuracy as a proxy for that this pathway, seems to be involved in this process. So when you're trying to identify this, rather than just say that there's a p-value, right, which is a perfectly reasonable thing to do, but rather than just say, here is the p-value, p-value tells you that on average these things are different and to what level of certainty you can say that, right, which is valid. But a lot of times people in the community take a p-value and they turn it into an accuracy when they start talking about it with others. So you'll get a p, right? So you see this happen a lot, right? There's a p-value and it says this pathway is enriched in prostate cancer, and then that becomes a feature of, the prostate cancer, for example, and so this just asks the simple question: okay, but if all I knew about this particular samples was the state of this MAP kinase network, to what degree, what accuracy would I expect that would in fact be true? Is it 90%? Right? You can have 60% accuracy and have a very good p-value if you've looked at it a lot of times. Right? If you have a lot of samples. And so that's sort of the idea that I think is important here. Okay, we've also looked at this in terms of just looking across. This is going back to GIST that I brought up at the beginning, and now asking the question, if we look at these networks or pathways in a different way and just ask, how similar is this template ranking between different individuals? So here we have highly regulated. So this means that 27 of the individuals here had exactly the same ranking. 6, this is G protein signaling pathway. And then you have this sort of set out here that are very different. This is sort of an intermediately regulated and a loosely regulated. And you'll see in each case we have sort of a set out here near the edge that are very different. And so the interesting question here is, are these outliers the same samples when we look across different pathways? Essentially, the question is this individual have a really abnormal behavior of a network, or is this patient as a whole, or this sample as a whole, very different from the other samples? Okay, so it's just which direction does that distinctness come from? And the answer here is mostly yes, they are the same when looking across all pathways, which means that it's mostly that the sample is really different. Okay, so we don't see that often localized differences in terms of one particular pathway that's very different for a particular individual, but the rest of what you look at is really similar. It's mostly that the individual is different. And so I think what's interesting here in terms of high throughput data and thinking about this is that when we do diagnoses, is to think about a core diagnosis versus a peripheral diagnosis, right? So in some of these cases, so if we were looking at these patients here, for example, it's like, okay, you have a gist, and it's very obviously from the molecular characterization that it really looks a lot like a gist. These other people here, maybe it's like, okay, this is a gist because that's the closest thing we know to call it. But molecularly is actually quite distinct, right? We might not have a really good sense for what this is. And this is one of the things I think is really interesting to think about in terms of how do we map this really globally as we're doing lots of diagnoses across patients. And as this kind of data really accumulates, I think we can start to get a sense of this landscape, right? So there's a disease landscape that we can imagine, and there are certain attractor points, right, which we call diseases that share similarities and that we treat. But in the peripheries, there's all kinds of other changes that we have to assign, sort of as close as possible to what we are, to a diagnosis that we can make, but that may in fact be something quite distinct. And so I think that's another important point. Okay. So I'm trying to see a clock because we had a lot of questions and I don't know where we're at. 5 minutes? Okay. Thank you. Okay, this is my last section anyway, so I'm about done. So the final story that I wanted to talk about was just to introduce some spatial gene expression analysis that we've done recently from the Allen Brain Atlas. So for those of you who don't know, the Allen Brain Atlas, which is just situated right near us there in Seattle, has done a really remarkable and amazing thing, which is to measure at very high, even single-cell resolution, the expression of genes in the mouse brain. Okay, and so this was a $100 million project funded by Paul Allen. He just recently gave them another $300 million, I heard. I hope that's been announced. And so they've had a lot of investment that's gone on to being able to build really a unique resource in the world. So this was the Allen Brain Atlas, as I mentioned, so that's just in situ hybridization. I hope these come out okay. And they've put online basically for anyone to use and given us a way to download all of this data such that we can do analysis, and anyone in the community can. So it's really a tremendous resource. So what we did is we took that data and then fused it with another data source, which is from John Cahoy's lab at Stanford, where they had done cell sorting of astrocytes, neurons, and oligodendrocytes to figure out which genes are expressed only in those particular cell types. So coming up with cell type specific transcripts. And so the idea that I wanted to look at here was, could we look at cell type specific transcripts and how they might define signatures in the Allen Brain Atlas. So we got neuron enriched genes, about 170 genes from about 300 different images that they had, oligodendrocytes, which was about 75 genes, and astrocytes, which is about 90. Okay, so if you just see, these are kind of characteristic images. I don't know if you can see that terribly well, but this is for neurons, just to give you a sense for what these look like. So you just have all these different expression patterns, and these are for genes that John Cahoy found to be specific to neurons. Okay, so then what I did here was to take all of the images, and this was done by Yoonhee Ko, who's a graduate student in my group, and essentially took all the images for neuron-specific genes and then just stacked them. So just do image registration, stack them up, and then we cut up each of the images into 10,000 patches. And so you have 10,000 patches and you have 300 observations, so you have a 10,000 by 300 matrix on which to do classification, essentially. Okay, and so what we did then was to do k-means clustering. Okay, so k-means clustering is a very simple approach to basically come up with clusters in this high-dimensional space. And so if we take this for the neuron-specific genes, and we just do clustering here, and we do 3, so 1 is just 0, the background, so there's only 2 here that are biologically meaningful. It tells us that the dominant gene expression difference in the mouse brain is cortex versus the rest. Okay, and you can see that very clearly, very cleanly. Not a surprising result, I suppose, but very interesting to me at least, like how unbelievably clearly it comes out. If you jump up the next number of clusters to 4, now the caudoputamen pops up. So this is now the 3 dominant differences between gene expression. You can go to 5, then the global pallidus pops up. 6, the cerebral cortex now starts to be striated, and you can see it there. The piriform cortex then pops up, which is right here, and so forth. And so the interesting thing here is that gene expression in the mouse brain is extremely well spatially correlated, particularly for genes that are specific to neurons. So if you look at them just through this lens of the neuron-specific genes and the beautiful project that they did at the Allen Brain Atlas, you start to see a very exquisite spatial patterning. In fact, we've taken this all the way out to 70, and you can see that here. Even with 70 different clusters, you can start to see very fine levels of the layers here in the cerebral cortex and throughout. And so you get just all kinds of very interesting patterning. And what this gives us back then are signatures at the gene expression level that are specific to each of these different subregions. And so this, we think, is really exciting going forward, just to think about, how precise molecularly or how far down can we actually go to be able to have signatures that relate not just to an organ, but even to subparts of an organ. Interestingly, if we take the genes that are oligodendrocyte enriched, you can see that there's clearly spatial patterning, but much less fine than you see for the neurons, right? So even though you can see this, some of this sort of spatial patterning, in all the genes, it's when you pull out that neuron subcomponent that it really pops into something that's really clear. If we do it for astrocytes, same thing. Of course, you get enrichment in the cortex here, but the caudoputamen sort of showing up a little bit here. But again, nothing like you see when you look at the neuron-specific genes. So this is also, I think, good evidence of the fact that, I talked in the blood about this notion of organ specificity we're interested in here. This is sort of analogous to cell type specificity, but by looking through this massive amount of data at a specific subset that is cleaned up to be just coming from one particular cell type, you can get a lot of enrichment. Okay, so just to sum up, I'll just sum up a little bit of what I talked about here today. We started by just introducing the simple notion of a relative expression reversal and how that can be used as a classifier. Then talked a lot about global analysis across the major diseases of an organ system. These are, I think, very informative things to look at. There's a lot of messiness associated with them since you're pulling lots of data from disparate studies together. But I think it teaches us a lot about some of the pitfalls that we have ahead of us in terms of being able to really take these signatures all the way through to the end. Introduce this Dirac method, which is a way for identifying perturbed pathways, again, based on these relative expression reversals. And then just to sum up, we went through the case of cell type specific genes and how regionally and spatially coordinated they can be. And this, I think, is very interesting as we think about diagnostics going forward, which is the level of specificity to which it may ultimately be possible to go. So with that, let me just thank people in my lab for their work. James Eddy developed the Dirac method. Yoonhee Ko did the analyses with the Allen Brain Atlas, and Seth Omics had also a big role in that particular project. Andrew Magis did the studies with colon cancer, looking at lab effects and doing the PCA analysis. And Jay developed the Isaac method and did the work with the brain specific classifiers. Oh, and I forgot one person I should mention, Shuyi Ma did also, who's another grad student in the group here, who's funded on an NSF fellowship to cite her funding. She did the slide on the lab effects in lung disease. And thank my funders for this as well, and happy to take any other questions. Thanks. Yeah.
One question.
Okay. So maybe you teach us another lesson. Maybe if these identified differences are real and we think about that, maybe it would be, we have to change our thinking about biomarkers for different regions. If they are real across different regions, I understand that it's convenient to count this one colon cancer in different region and to have only one marker. But maybe we have to think about different set of different biomarkers for different regions based on what you showed us.
Yeah, I think it's absolutely a great point. And so, you know, one of the things, and we're trying right now to build sort of a big automated system that will sit on top of Geo and sort of run these kind of things. But one of the things that I think is so interesting is exactly the point you bring up, which is, okay, we start overcoming lab effects, we start seeing you know, classification accuracy stabilize. But when they're lower than what we think, you know, than they need to be, I think it's really about this. How do we get into the heterogeneity? And all these factors start to matter. Like, where in the brain did it come from? I mean, obviously that has a huge impact. And yet in most of the studies that we did in that first one with the brain, it wasn't even annotated. We wanted to take that into account, but it wasn't annotated. You know, oftentimes we don't know precisely where it came from or what region it came. Sometimes we do, but a lot of times we don't. And so all of that hidden information is probably going to be necessary to attack the. So when people, you know, when we start talking glibly about, you know, omics data, and then okay, we're going to get signatures, and then we're going to be able to read out, you know, health states. It's like, okay, eventually, yes, I totally believe that is true, but there is a lot of complexity to deal with in between. And so I think we're just getting into more and more exciting times. As we're starting to get the sample size numbers we need, we're starting to have the amount of information we need. So from my standpoint, you know, I think we should be going great guns in this area, but it is complicated. It's going to take a while to get there. So yeah, it's an excellent point.
Thank you so much, Nathan.